Hello everyone, welcome to Jnanataranga program, CET coaching classes for the year 2018 and 19. This program has been organized by Department of Pre-University, Karnataka. I am Sulakshana, lecturer in biology, Government PU College, Pavgada, Tumkur. So today we are going to discuss about the topic cell structure and function, that is belonging to the unit cell, the unit of life. So in this topic we are going to study about the details as follows. First of all, regarding the cell theory. Cell theory was proposed by Schleiden and Schwann, and it was improvised further by Rudolf Virchow. And it says that all the organisms' body contain cells, and all new cell can arise from the pre-existing cell. And comes the cell types. Cells are classified based on their nuclear arrangement into prokaryotic cell and eukaryotic cell. Prokaryotic cell has got following structural components: cell envelope, cellular outgrowth, and cytoplasm. Cell envelope is a component which is made up of three layers: glycocalyx, cell wall, and plasma membrane. Glycocalyx is containing slime layer as well as capsule. Cell wall is a layer made up of murin or peptidoglycan, and based on the chemical composition which can be classified into gram positive and gram negative. And the third component is plasma membrane, and plasma membrane shows inward foldings called as mesosome. Next to it is the cellular outgrowths, which are referred as pili, fimbriae, and flagella. Pili and fimbriae are the outward growths which are proteinaceous in nature. Pili is found only in gram negative bacteria, and it is made up of protein called as pilin. Pilin is encoded by the genes present on the plasmid DNA, whereas fimbriae is made up of protein called as fimbrilin, and their genes are present on the DNA of nucleoid. And third comes the flagella. Flagella are the motile structures, or the structures helping in motility, and they are quite longer or larger structures when compared to pili and fimbriae. Lastly, the cytoplasm. Cytoplasm is containing mainly nucleoid, that is a genetic material, and plasmid. Plasmid may or may not be seen in a bacterial cell, and 70S ribosomes, polysomes, and inclusion bodies. And all other membrane-bound organelles are absent in a prokaryotic cell. Eukaryotic cell is basically classified into cell wall. Cell wall is found only in plant cells and few other organisms, and if at all a plant cell is considered, cell wall can be classified into middle lamella made up of calcium and magnesium pectate, primary wall made up of cellulose, hemicellulose, and pectin, and secondary wall it may be made up of lignin or suberin. Next component is protoplast. So protoplast is the entire living content within the plant cell, or it is generally a typical animal cell. When protoplast is considered, it has an outer covering living membrane called as plasma membrane. In plants you can find vacuole, which is surrounded by tonoplast, and it contains cell sap. Then in between the plasma membrane and tonoplast you can find the cytoplasm. So cytoplasm further holds the organelles, cytoskeleton, and ergastic substances. Organelles as discussed in detail, but cytoskeleton is the structural framework made up of proteins or microfilaments which provides the stability to the cell, and it will help in cytoplasmic streaming or sometimes even in the process of cell division. Next comes the ergastic substances. Ergastic substances are the non-living inclusions found only in plant cell, which can be including the starch granules or the mineral crystals. Now let us discuss in detail about the organelles. Cell organelles of a typical eukaryotic cell can be classified into membraneless organelles, membrane-bound organelles. Membraneless organelles include ribosomes, which are generally referred as the protein factories of the cells. They are considered as 80S ribosome if they are present in the cytoplasm. and they are classified into larger and smaller subunits. The larger subunit of 80S ribosome will be 60S subunit, and the smaller unit will be referred as 40S subunit. Further comes the centrosome. Centrosome is a complex made up of centrioles and the centrospheres. Centrosphere is a special location of the cytoplasm where the centrioles are present. Centrioles are the cylindrical, membraneless, proteinaceous structures occurring in pair, and they are perpendicular to each other. Moving on to membrane-bound organelles, they can be classified into membrane depending on the number of membranes, like double membrane-bound organelles and single membrane-bound organelles. Double membrane-bound organelles mainly include nucleus, mitochondria and chloroplast because they are surrounded by two separate membrane systems, and single membrane-bound organelles include endoplasmic reticulum, Golgi complex, lysosome and microbodies. Here comes the questions regarding the same topic. The question goes so: which of the following is not true for a typical cell? The options are as follows. The cell is a fundamental structural and functional unit of life. Unicellular organisms are capable of independent existence with capacity to perform essential functions of life. C. Structure of nucleus is mandatory to hold genetic material of any cell. D. Presence of non-living outer cell wall is clear in kingdoms like Monera, Mycota and Plantae. So out of the four options, the statement which is not true is statement C. Structure of nucleus is not mandatory, or it is not a compulsory structure to hold the genetic material, because nuclear structure is visible only in eukaryotic cells. Further, if you consider prokaryotic cells, their genetic material just lie in the cytoplasm, free in the cytoplasm in the form of nucleoid. So answer is C. Second question: the striking difference between animal cell and plant cell is that A. Animal cell is devoid of cell wall. B. A large area inside the animal cell is occupied by vacuole. C. Chloroplasts are common organelles found both in animal and plant cell. And D. Animal cells are eukaryotic in nature. So the question is slightly tricky because the question asks the difference between animal and plant cell. So what is very special about animal cell when compared to plant cell? So the answer is A, that is, animal cell is devoid of cell wall, or they do not contain cell wall. Remaining statements are actually proving wrong because animal cells, as mentioned in option B, do not contain vacuole. It is the feature of plant cell. Option C says that chloroplast is present both in animal and plant cell. So it is very clear chloroplasts are found only in plant cell. Option D, it says only animal cells are eukaryotic. Actually, all the organisms belonging to four kingdoms except Monera are eukaryotic. So Protista, Mycota, Animalia and Plantae, all these four kingdoms include eukaryotic cells. So answer for second question is A. Here comes the third question, that is to match the contents of the table. Table A indicates the names of the scientists and table B indicates the contributions of those scientists. So first comes Anton von Leeuwenhoek, second George Palade, third Robert Brown and fourth Camillo Golgi. And the contents of the table B are discovered nucleus, second one observed dense reticular structures near the nucleus, or they are referred as Golgi apparatus. Third one is observed and described a live cell. and fourth content is first to observe ribosome under electron microscope. So the options are mentioned below. So what you can observe here is there are 4 options. The options run like this. Option A 1, 2, 2, 3, 3, 4, 4, 1, and option B, C, D are running in the same way. So let us try to understand the contributions of every scientist there. Once if you continue with Anton von Leeuwenhoek, we already know that he was the first one to observe and describe a live cell. So for first one, option is Roman number 3. Second scientist, that is George Palade, was the one to first observe ribosome under electron microscope. For the reason same given there, ribosomes are also referred as Palade particles. Option C, third one, Robert Brown. We all know that he is the discoverer of nucleus. And fourth one, Camillo Golgi, as his name indicates, he is the one to observe the Golgi apparatus. So the answer which goes right here is option B. Moving on to fourth question. What is common between chloroplast and mitochondria? Options come here. Both are semi-autonomous organelles. B, presence of 70S ribosome. C, ATP synthesis. D, all these. In the previous synopsis sheets, we have understood that the common nature between chloroplast and mitochondria is that both of them are double membrane bound organelles just like nucleus. Now we can call them as semi-autonomous organelles because in their matrix, or what you call as stroma, they have got circular double-stranded DNA which is capable of carrying out the division or multiplication of mitochondria and chloroplast without the instruction of the nuclear DNA. Second option is presence of 70S ribosome. Yes, though these two organelles are found in eukaryotic cells, within these organelles we can find 70S ribosomes just like as in bacterial cells. C, we can see ATP synthesis in both these organelles. Mitochondria will go for the process of ATP synthesis called as oxidative phosphorylation, and chloroplast will synthesize ATP by a process called as photophosphorylation. So this question has got the clear answer, that is D, all these. Moving on to the fifth question. A typical cell membrane or biomembrane has all these except. So the question wants us to understand the chemical nature of the plasma membrane, as according to Singer and Nicholson, who proposed fluid mosaic model. We have seen that plasma membrane is containing 2 layers of phospholipids. That's why it is referred as bilipid layer. And in between these lipid molecules you can find different kinds of proteins classified into intrinsic protein, extrinsic proteins and transmembrane proteins, and you can also see the chains of cholesterol. And then we have got different kinds of sugars classified into glycolipids and glycoproteins. What is not found in plasma membrane is option B, proline. Proline is an amino acid, so it is not directly the part of a plasma membrane. Next comes the table which indicates the organelles in the first position, and the middle table indicates their structural details, and the third table indicates their functions. Now we are supposed to match them. Vacuole is the first option there, then comes lysosome, third one endoplasmic reticulum, fourth one is the cytoskeleton, and in the middle table we have got they are derived from Golgi bodies, next network of filamentous proteins, next tonoplast, and the last one is continues with the outer nuclear membrane. And in the last column we have got the functions. The first one goes like this: holds water and excretory products. 2. Helps in protein synthesis and secretion. 3. Contains the digestive enzymes. 4. Maintains the cell shape. So let us match them now. Vacuole is actually associated with the tonoplast, so 1 goes with R. And if you have to look into its function, in the third column, we can say that it holds the water and excretory product. So 1, R and Roman number 1 matches very well. 2. Lysosome. It is usually said that it is derived from the vesicles of the Golgi body, which will be filled with digestive enzymes. So for second one, the options goes like this: 2, P and 3. Third one, endoplasmic reticulum. We have observed the position of endoplasmic reticulum, that it continues with the outer membrane of nucleus, and if you consider the functions of it, it will help in protein synthesis and secretion. So endoplasmic reticulum 3, S and 2, all these will match. And with respect to its function, I just want to give one clarification. Endoplasmic reticulum are associated with ribosomes. If it is so, we call it as rough endoplasmic reticulum. If they do not contain any proteins on their surface, they are called as smooth endoplasmic reticulum. Rough endoplasmic reticulum, because it is associated with the ribosomes, it is associated also with the function of protein synthesis, and the smooth endoplasmic reticulum will go for secretion process. The last one is cytoskeleton. Cytoskeleton is network of filamentous proteins, and their function is mainly to maintain the cell shape. So the option for this particular question is D. Moving on to the next question. The classification of bacteria into gram positive or gram negative is based on the charge carried by the bacterial cell, the density of the cell, response to specific staining technique, or the presence or absence of plasmid. The right option for this, or the right answer for this, is option C, which is the response to specific staining technique. Now let me give some detail of this option. The process of staining, which concludes a bacterium whether it is gram positive or gram negative, is based on a technique called as Gram's staining. This technique uses crystal violet dye, which will dye or color the outer layer of the bacterial cell. Further, depending on the amount of phospholipids as well as certain components of peptidoglycan, you can consider the difference in the colors. So the bacterium which has got more peptidoglycan and less amount of lipid will be showing the purple color, and you consider those bacterium as gram positive. Gram negative bacterium is those which has got less peptidoglycan and more lipid, and after counter staining it appears pink. So if all the bacterium in a slide shows purple color, they are all gram positive. If they show pink color, they are gram negative. So the option for 7th question is C. Moving on to next question. The mentioned set of functions are exhibited by which of the following cellular components? So the functions are listed in the bracket: active and passive transportation, protection, and selective permeability. So these are the functions associated with the transportation, providing protection, and being selectively permeable is the property of plasma membrane, whereas nucleus, ribosome and centriole, at least these cannot provide protection. For providing protection, it has to be on the periphery. So this main function may give us the particular clue for the answer. So option is A for this question. Next question: Which of the following statements is not true for nostoc? Nostoc is a prokaryotic blue-green algae. It is photosynthetic. It checks soil erosion and improves the soil fertility. It is branched and multicellular algae. So let us get into the details of nostoc. Nostoc is considered to be the filamentous algae having a mucilaginous sheath or mucilaginous covering, and the first option is very true because nostoc is belonging to cyanobacteria of kingdom Monera, so first one is right. B, photosynthetic. Yes, it is blue-green algae. It has got photosynthetic lamellae with specific pigments, and this is the first organism to carry out oxygenic photosynthesis. That is, the photosynthesis reaction releasing oxygen as the byproduct. C, it checks soil erosion and improves soil fertility. That is also true because the mucilaginous sheath of nostoc will hold the soil particles together, thereby it avoids the soil erosion. And of course, it improves the soil fertility also because it is containing a specific cell called as heterocyst, which with the help of an enzyme called as nitrogenase will fix the atmospheric nitrogen. The last option, D, is actually the right option. It is not the true property of nostoc because nostoc is unbranched and it is unicellular algae which is colonial or lives in groups. Moving on to next question. Which of the following sets of components rightly indicate the details of a prokaryotic cell? First set indicates plasmid, glycocalyx, mesosome and pili. Second one, plasmid, glycocalyx, mitochondria and pili. Third, 70S ribosome, slime layer, flagella and nucleus. D, nucleoid, plasma membrane, chloroplast and polysome. So the question asks which are the components which are found only in prokaryotic cell. So option A is the right one because all the four components are the parts of prokaryotic cell. In option B, mitochondria is not the right option. In C, because of nucleus you cannot include this option, and in D, because of chloroplast this is not the right combination. So for 10th question, answer is A. Next comes the diagram question where we are supposed to indicate the labels. The diagram is representing animal cells and the labels are A, B, C, D. The options given there are A, microvilli, B, centriole, C, mitochondria, D, RER standing for rough endoplasmic reticulum. B option has got cilia, cytoskeleton, mitochondria and RER. For C, we have got microvilli, cytoskeleton, chloroplast and SCR. For D, microvilli, centriole, chloroplast and Golgi complex. So the answer for this one is, we can clearly see it there. The foldings on the surface of the animal cell which increases the surface area for absorption or any other activity is referred as microvilli. So option A is microvilli. B, the labeling is indicating certain paired structure which are in close association with the nucleus. They are nothing but the centrioles. Option C is indicating the capsule-like or elliptical organelles which are having the convolutions or foldings in the inner surface. So it indicates mitochondria. And the last one, D, is indicating the lamellar structures or intracytoplasmic reticular structures holding the ribosomes on their surface. So it is RER or rough endoplasmic reticulum. So the option goes right with this diagram is option A, microvilli, centriole, mitochondria and RER. Next question, identify the mismatch. The mismatch indicates The organelle as well as their functions are indicated there. So let us identify the mismatch or wrong combination. The first one, A, lysosome, and it says hydrolytic enzymes. It is very true because lysosomes are filled with digestive enzymes or hydrolytic enzymes which will help in various processes starting from digestion up to a certain function of immunity. B, centriole, associated with cell division. This is also the right combination because from the centrioles, asterase and spindle fibers are formed which will help in the process of cell division. C, Golgi body, it is indicated as protein synthesis. Now, protein synthesis exactly is not the function of Golgi bodies. Protein synthesis is directly associated with ribosomes or the rough endoplasmic reticulum which on its surface has got the ribosome. Golgi body has nothing to do with this particular function. Its main function is secretion, slightly transportation, detoxification as well as packing of biomolecules. And D, vacuole, it helps in osmoregulation. This is the right combination. Osmoregulation is regulating the amount of water within the cell. So vacuole does it for plant cell exactly. So in this 12th question, the mismatch is option C. Golgi body and protein synthesis are not the right combination. Moving on to next one. Which of the following statements regarding nucleus is not true? Nuclear material stained by basic dye like methylene blue as observed by Fleming. B, it has nucleoprotein fibers called chromatin and dense non-membranous spherical body called nucleolus. C, like mitochondria and chloroplast, nucleus is also double membrane bound. Option D, outer nuclear membrane is smooth and inner membrane is convoluted. So out of the 4 statements, we have to identify the statement which is not the right one. So first option indicates that a scientist Fleming, he made use of a dye called as methylene blue to color the nucleus, and nucleus is containing the DNA, therefore it is acidic and it is rightly stained by a basic dye. So option A is correct. Option B is reading like this: nucleoprotein fibers called as chromatin is present, that is DNA, and the membrane-less structure is nucleolus. Option C is right because all these 3 organelles including nucleus are double membrane bound. And moving on to the 4th option, outer membrane is smooth and inner membrane is convoluted. Convoluted means folded, but it is only the property of mitochondria. So the statement which is not right is option D. So in mitochondria, inner membrane is convoluted to form structures called as cristae. So this property D is associated with mitochondria. So answer for 13th question is D. Moving on to 14th question, we have got sets of chromosomes here, different types of chromosomes classified based on the position of centromere. So question is, by looking at the following representations of the chromosome, name them in the same order based on the position of the centromere. The first diagram has the centromere exactly at the center. Second one has got it slightly away from the center, as a result of which one of the side of the arm looks shorter and the other side of the arm looks longer. Third diagram indicates once again the centromere away from the center, almost near to the end, and fourth diagram is having the centromere almost at the tip with a very short arm on the upper side. So this set of diagram indicates the chromosomes can be classified as metacentric, submetacentric, acrocentric and telocentric respectively. So the options here are the B one. The option B is the right one. First chromosome is metacentric, second one is submetacentric, third one is acrocentric and fourth one is telocentric chromosome. The next question is regarding the structure of chloroplast. Identify the functions of the part. Actually, it should be the labeling given there. So, labels are A, B, C and D. Option A is actually the membrane. It indicates the label called as membrane. B label indicates the grana, that is the set of thylakoids there, and option C indicates the stroma. So, the right answer for this question is option B. Moving on to the next topic, biomolecules. Biomolecules are classified into biomicromolecules and biomacromolecules depending on their size. So, under biomicromolecules we have got lipids, and biomacromolecules are all the polymers referred as proteins, nucleic acids and polysaccharides, and all these 4 components are present in the acid insoluble fraction when the separation of chemicals of a tissue are done. Details of lipids. So, lipids have got molecular weight of less than 1000 Dalton. It is water insoluble, therefore we call it as hydrophobic. Then comes, they could be simple fatty acids, glycerol or both. So, they can be classified further into monoglycerides, diglycerides and triglycerides. They can be classified as fats and oils based on melting point, and the last classification is the group called as phospholipids, where the lipids are associated with the group of phosphorus. Next comes proteins. Proteins are considered as the polymers of amino acids which are linked by peptide bonds. Structure of proteins can be classified into primary, secondary, tertiary or quaternary. Primary structure of protein indicates the straight line of amino acid or linear arrangement of amino acid, and the examples coming under these are insulin, myoglobin and lysozyme. Secondary proteins are the one where the amino acid chain or the polypeptide chain is coiled, or it shows the helical structure. You can include the protein called as keratin here, and keratin is the very basic protein which we contain in our hair, nails or animals have it on their hoof. Tertiary proteins are the one where the polypeptide chains are spherically coiled or it forms a ball like structure, and you can include globular proteins under this. And the fourth classification is quaternary protein. Quaternary proteins are made up of more than 2 polypeptide chains which are associated with each other. Say for example hemoglobins. Hemoglobins are containing 2 polypeptide chains indicated as alpha chain and beta chain. And the last function of this is also mentioned. It is a special function and very important function of protein. They also act as enzymes. Next polymer is nucleic acid, which we considered as the genetic material, and these are the polymers of nucleotides which are connected by phosphoester bonds. So, the types of nucleic acids are DNA and RNA, and they basically contain all the kinds of nucleic acids are made up of sugar, phosphoric acid and nitrogen bases. The last category of biomacromolecules are polysaccharides. So, polysaccharides are the polymers of sugars. They contain monosaccharides which are linked by glycosidic bonds. Next comes enzymes. So, enzymes are the proteins acting as biocatalysts. They can be classified into following groups, though they are oxidoreductases, which are referred as dehydrogenases. Next category is transferases. Third one, hydrolases. fourth one lyases, fifth ligases, and sixth one isomerases. All this categorization is merely depending or based on the functions carried out by this group of enzymes. Now, the factors that affect the activity of enzyme or the rate of enzyme action. The main factors that affect the rate of enzyme action include temperature, pH, substrate concentration, and different kinds of inhibitors, specifically the competitive inhibitors. Now comes the questions. So in this question, we are having two statements and we have to conclude based on the concept given there. Statement A goes like this: primary metabolites play key role in normal physiological processes, while secondary metabolites are not associated with basic physiology. Statement B: however, secondary metabolites are ecologically important for the survival of the plants and also useful to the humans. So here what we can understand is that in case of plants, we can classify their chemicals into primary metabolites and secondary metabolites. Let me give the clarification. Primary metabolites are the chemicals which are very important to carry out the very basic functions of a plant body. Secondary metabolites are the derivatives of primary metabolites and they carry out specific functions in plant body, like providing protection or avoiding the predators, avoiding the grazing animals, or different kinds of infections. And these secondary metabolites can be including the types like alkaloids, phenolics, tannins, etc. And because these are having the antimicrobial property and different kinds of physiological functions, humans also use them as medicine. Say for example, vincristine and vinblastin associated with the plant Vinca rosea. These two chemicals, I repeat, vincristine and vinblastin, are having anti-cancerous properties. Same way, the plant Sarpagandha or Rauwolfia serpentina has got the chemicals called as reserpin and serpin. Both of them are associated in regulating the blood pressure of humans. So as a result of this, we can come to a conclusion that both the statements are correct. So option for this question is A. Next one is matching the contents. So in the first table we are having the proteins, different kinds of proteins, and in the second table we are associating them with their function. Antibody, we all know that it is associated with providing protection to the human body or animal's body. So it goes with option 4, immunity to organisms. Next comes insulin. Insulin is a proteinaceous hormone secreted by pancreas and it is required for maintaining the blood glucose level. So option 1 goes very well with this. Next comes trypsin. Trypsin is a digestive enzyme. And last one, collagen, it is a binding material found between the animal cells which holds all the cells together. So the option for these matching tables is option B, that is P4QIR2S3. Moving on to next question. Here comes the details of different types of proteins found in biosphere. So let us discuss the detail. Abundancy of which of the following proteins is more in biosphere? So biosphere has got which of the following protein in large quantity? The options are collagen, rubisco, palmitic acid, and cellulose. The answer for this question is B, that is rubisco. So Rubisco is an enzyme, therefore a protein, which helps in the fixation of carbon dioxide during the process of photosynthesis. So this is the protein which is having large abundance in biosphere compared to all other protein. Option A, collagen, though it is a protein, it is the most abundant protein found only in animal world. Option C, palmitic acid, it is actually a saturated fatty acid found in all the kinds of lipids, so they are found in animal's body. And option D, cellulose, cellulose is most abundant organic molecule, not only protein. They are specifically answering about the protein, but about cellulose we have to remember that it is the most abundant organic molecule. So option for this question is B. Moving on to next question, fourth one. Which of the following pairs do not indicate polysaccharide? So we know that polysaccharide is the polymer of monosaccharides or the sugar units. The pairs goes as follows. First one, starch and glycogen. Second, chitin and cellulose. Third, inulin and leaven. D is lecithin and myelin. So we have to identify only the pair which is not polysaccharide. So just remember, starch and glycogen are the storage polysaccharide found in plants and animals respectively. So both of them are storage polysaccharides. Option B, chitin and cellulose, these are referred as the structural polysaccharides which builds up the cell wall of a specific organism. So chitin can be found in either in mycota or in insects, and cellulose is the cell wall material of plant. Third one is inulin and leaven. These are the polysaccharides derived from fructose. Last option, lecithin and myelin, these are not polysaccharides at all. They are actually the lipids, different kinds of lipids referred as a lecithin and myelin. Just remember, lecithin is found in soybean and egg, which are important in understanding the working of the memory associated with the brain cells. So whenever a person is associated or having disorders of memory like dementia or Alzheimer's disease, they are administered with lecithin. So lecithin helps in building the memory. Whereas myelin is the fat or lipid which is present in the axon of the nerve cell where it provides the protection. For the fourth question, the right option is D. They are the pairs of lipids, not polysaccharides. Fifth one, identify the statement which is not true with respect to nucleic acids. They are polymers of nucleotides linked by phosphoester bonds. Heterocyclic compounds of nucleic acids are purines and pyrimidines. C. Nucleic acids are found in acid soluble fraction of living tissue. And option D, its sugar is pentose and also referred as furanose. The answer, the right option for this question is option C, where nucleic acids are found in acid insoluble fraction of living tissue. So these are not the acid soluble component, they are the acid insoluble component. Therefore, C option is the right one. Sixth one is indicating the structure of DNA, and we have labeled the different bonds present in them. We have to identify the different bonds now. Label A, Label A is indicating a bond extending between the pentose sugar and phosphate group. We call it as phosphoester bond. Option B, or sorry, the label B. Label B is indicating a bond between the pentose sugar and the nitrogenous base, that is extending from the first carbon. We call this as glycosidic bond. And the third C label is indicating the bonds between the two nitrogenous bases like guanine, cytosine. This is indicating the hydrogen bond. So A is phosphoester bond, B is glycosidic bond, C is hydrogen bond. The right answer for this question is option A. Moving on to the next one, 7th slide. 7th question is indicating a graph which explains the concept of activational energy during enzymatic reaction, and we are supposed to identify the labels here. So label A is indicating the peak of this graph. Label B is indicating the last point of this graph, and the label C indicates the arrow mark extending from the median line to the topmost point. D is indicating another level here. So just remember, this graph is indicating the following component. So the graph here is having the substrate which is entering into the reaction, and the graph is indicating two conditions. The graph which is having a highest peak is associated with the rate of reaction and potential energy required for that when a reaction is not making use of the enzyme. And this particular point here labeled as A is indicating the transition state, that is a temporary unstable state formed by the association between substrate and the enzyme molecule. And when at the end you consider the position, we have got B here, this indicates the product, and the arrow mark C is indicating the amount of activational energy that is required when a reaction is not associated with enzyme, and the arrow mark D is indicating the amount of energy required when the reaction is making use of an enzyme. So as a result of this, we can conclude that a reaction where enzyme is used as a biocatalyst will require very less amount of energy to complete the process. So as a result of this, we always come to a conclusion that enzyme related reactions are faster, products are quickly produced, and energy required for this reaction is very, very less. So options given here are 4, and label A should be indicating, as I explained, transition state. Label B should indicate the product. C should indicate the activational energy without enzyme. D should indicate activational energy with enzyme. So option A is the correct one. Moving on to next slide. Question number 8. Below reaction represents the enzymatic reaction and formation of product. ES here stands for enzyme substrate complex. And what are their properties? So now we are supposed to explain the properties of ES complex. So the reaction is represented there. E is enzyme, S is a substrate, ES is enzyme substrate complex, EP is the temporary association between the enzyme and the final product, and last one, E plus P indicates the separation of enzyme from the final product. So the options are as follows. ES is a product which is unstable. ES is modified form of enzyme which forms the substrate. It is a stable product of enzyme action. ES is short-lived. highly reactive intermediate of any reaction. So what we can understand here is ES is not a product as explained by the first option. So first option is wrong. B option says that it is a modified form of enzyme which forms the substrate. So it is indicating the reverse of reaction. Always we have to think of formation of product. So B statement is wrong. C, it is a stable product of enzyme action. So it is never a stable product, it is a temporary highly reactive stage. So the option that goes well with this question is ES is short-lived, that is having a unstable condition. It is highly reactive, intermediate, meaning to say not the final product. So it is the intermediate of any chemical reaction. So option for this question is D. Next question. The narrow range of temperature at which enzyme shows highest activity is called optimum temperature. Which of the following statement regarding it is not true? So we all know that temperature is one external factor which will affect the rate of reaction by affecting the enzyme. So let us see what statement is wrong with this. First option is change in temperature alters the tertiary structure of protein or enzyme. B, low temperature preserves the enzyme in temporarily inactive state. C is if it is a stable product of enzyme action, and D, ES is a short-lived, highly reactive, as well as option C goes like this: higher temperature causes denaturation of enzyme, and option D, denaturated enzymes are efficient in carrying biochemical reactions. So the options are A, B, C, D, out of which we can identify this. Option A is true because proteins change their tertiary structure when they are exposed to different variations of temperature. When the enzymes are in low temperature, they become inactive. If it is exposed to higher temperature, it will undergo denaturation. Therefore, option D is not true. So the answer for this one is D, where denaturated enzymes are efficient in carrying biochemical reaction. This is actually not right. Denaturated enzymes cannot carry out the biochemical reaction. Moving on to 10th question. After Vmax in the below graph, the rate of reaction does not change. So let us understand the graph first of all. So lower component is indicating the substrate, and on the other axis we are having the velocity of reaction. So velocity of reaction and the substrate when rate of reaction when it is compared, we can identify that the graph is taking a leap here and it is coming to a stationary phase here, or it is becoming steady. So this particular graph is indicating the reaction is completed, or B, all the enzyme molecules are saturated, C, substrate concentration is less, or D, half Vmax is not yet reached. So what the graph is indicating is the steadiness of the reaction after reaching a particular level, or why does the velocity of the reaction does not change after reaching a specific level, is because all the enzymes are now saturated. Though the substrate is more and more, there are no free enzymes to attach with the substrate and to carry out the reaction. Therefore, the option for this particular question is B. All the enzyme molecules are saturated. Moving on to the next one, question number 11. If a molecule which closely resembles the substrate, as a result of which it does not allow the latter to bind with enzyme, the rate of the reaction will decline. Name the molecule responsible for this. So here we are including another factor of enzyme. Coming to the 11th question. If a molecule which closely resembles the substrate, as a result of which it does not allow the latter to bind with the enzyme, the rate of reaction decline. Name the molecule responsible for this. Okay, in this question we are discussing about one more factor which affects the rate of enzymatic reaction. Options given here are promoter, competitive inhibitor, regulator and non-competitive inhibitor. So be clear with this. Promoter and regulator are not associated with enzyme action at all. Therefore, those two are not the right answers. So exclude them. We are left with competitive inhibitor and non-competitive inhibitor. Just remember, inhibitors are the group of chemicals which will drop the enzyme activity because of their association with the enzyme. With respect to competitive inhibitor, what we have to remember is that it is a substance which will block the active site of the enzyme, as a result of which the substrate cannot come and bind to the enzyme molecule. Whereas non-competitive inhibitors are the kinds of inhibitor which associates to any part of the enzyme, whether it is associated with substrate or not. So answer for this particular question is option B. D is the right option, and D is not the one, because the question clearly says that the molecule should closely resemble with the substrate and it should not allow the latter. Latter means substrate to bind with the enzyme. So that molecule is competitive inhibitor. So answer for this is B. Next question. Along with the polypeptide chains, an enzyme may be associated with non-protein part. This part is called as. So while studying the structure of enzyme, we have got terms like holoenzymes, apoenzymes and cofactors. Let me give you a clarification. An enzyme is associated with the two parts. So one is apoenzyme, another part is the cofactor. Apoenzyme is actually the protein part of enzyme and cofactor is the non-protein part of the enzyme. So association of both, that is apoenzyme and cofactor, both of them will make up something called as holoenzyme, indicating that it is the entire enzyme. So entire enzyme has got protein part apoenzyme and non-protein part, that is cofactor. Now cofactors are having three separate types. They can be classified into prosthetic group, or they can be classified into coenzymes, and the third component is metal ions. So all these three are the types of cofactors. Do remember, cofactors, as I said, they are the non-protein part, where the prosthetic group is organic part permanently attached to the apoenzyme. They are tightly attached to apoenzyme, you cannot separate it. Coenzymes are once again organic, but their association with the apoenzyme is transient, that is temporary, which occurs only during the chemical reaction. And the third component or third type of cofactor is metal ion, which will be attached to the active site as well as to the substrate by forming the side chain. So do remember, the non-protein part that they have mentioned here is actually D, cofactor. Apoenzyme is the protein part, B, holoenzyme is the entire enzyme, having apoenzyme and cofactor. So answer is D for this. Next question is match the following type question, where we are having classes of enzyme on one side or groups of enzyme on one side and their function on the other side. So groups of enzyme: hydrolase, isomerase, ligase, lyase, oxidoreductase and transferase. Now we have to remember their function. Let us see the activity of hydrolase. Hydrolase is hydrolytic enzyme which will use water to break the chemical bonds between the components. So chemicals are broken or their bonds are broken by making use of water. Therefore, such enzymes are called as hydrolase. Moving on to isomerases. Isomerases are the one which will convert one form of the isomer into its another form. Third, ligases, as the name indicates, these are the enzymes which are capable of linking two molecules together. Fourth one, lyases. Lyases are associated with the function of joining the molecules or the groups by forming the double bonds. So they will remove the single bond and forms the double bonds. Next one is oxidoreductase. The name clearly indicates it is associated with electron. So option for this is the first one, that is, it transfers the electrons from one molecule to another, thereby causing the oxidation and reduction one after the other. The last one is transferases. We are left with this and the name itself indicates, so it will help in the transferring of functional group to another molecule. So from one chemical group it will transfer the functional group to the other one. So with the following function, the option that matches very well is option B. So answer for 13th question is B. Next one, moving on to the topic of cell cycle and cell division. So cell cycle is indicating all the sequence or series of changes that occurs in a cell from its formation until it divides into two daughter cells. So these processes are referred as cell cycle and we can classify them into different stages. So the stages are interphase and mitotic phase. Interphase is having the substages like G1 phase, S phase and G2 phase. Mitotic phase is further classified into karyokinesis and cytokinesis. Karyokinesis is subdivided into stages like prophase, metaphase, anaphase and telophase. And cytokinesis is associated with the division of the cytoplasm. At the end, we have got a special condition or stage referred as G0 phase or G0 phase. It is a special phase where the cell will enter into a non-dividing stage. So non-dividing cell will be always in G0 phase, but it does not indicate that it is inactive. These cells are metabolically active itself. Next one, types of cell divisions. So depending on different features observed there, we can classify the divisions into mitotic and meiotic division, or simply called as mitosis and meiosis. Now the components under these are as follows. So meiosis is having the stages like meiosis 1 and meiosis 2. Each of them are subdivided into prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinesis 1. Do remember, out of all the stages of meiosis, prophase 1 is the longest phase. It takes a very long time, having the sub-stages, and cytokinesis 1 is not a mandatory stage. That means after completing meiosis 1, a cell may or may not divide. In that case, it will divide into 4 daughter cells all at once at the end of cytokinesis 2. Meiosis II is also further classified into prophase II, metaphase II, anaphase II and telophase II, and ultimately the parent cell will divide into 4 daughter cells at the end of cytokinesis II. Moving on to the differences in the process of mitosis seen in animal cell and plant cell. So let us look at the differences here. Now, animal cells contain the centriole, therefore we say that spindle pole during mitosis will show the presence of centriole in animal cell, whereas plant cell do not have the centriole. Animal cell is referred as amphiastral. This is for the reason that when animal cell is undergoing mitosis at both the poles, because they are having the centrioles there, they form thin filaments or fragments of proteins which look like stars. This set is referred as aster ray. And remember, aster rays are found only in animal cells on both the poles. Therefore, it is referred as amphiastral. Amphi means both. Whereas plant cells, they do not have the centriole at all. Therefore, they do not show the formation of aster ray at the poles. As a result, because of the absence of aster rays, we call the process as anastral. Next, moving on to cytokinesis. Cytokinesis in animal cell is occurring with the help of furrow formation. So, furrow formation occurs at the periphery here, and the filaments, microtubules, all these will start pulling the plasma membrane towards the center of the animal cell, as a result of which the animal's cells will show the furrow or cleavage like this. When it comes to plant cell, because they have got an outer rigid wall, there is no furrow formation. Instead, they show the formation of phragmoplast at the center, which will be converted into cell plate later. So, cell division or cytokinesis is completed by the formation of cell plate. Next comes cytokinesis. The position is indicated. Here cytokinesis extends centripetally, whereas in plant cell it occurs centrifugally. It clearly indicates that centripetally means from the periphery to the center. So, therefore, it is centripetally. Whereas in plant cell, it starts from the center and moves towards the periphery. Therefore, it is centrifugally. The last difference is regarding the controlling of the process. In animal cell, chemicals referred as mitogens will control the process of mitosis, whereas in plant cell they are controlled by phytohormones or plant hormones called as cytokinins. Moving on to questions. The first one, quiescent stage where cells are metabolically active but do not proliferate is also called as, meaning is same, quiescent stage where the cells are metabolically active but they do not multiply or do not divide. So, it is indicating that the cells are active but they are not dividing. So, the answer for this is, as mentioned before, option C, because in G1 phase there will be preparation for mitotic division, all the proteins for DNA synthesis as well as cell division will be formed during G1 phase. S phase is a stage during which DNA doubling will occur, therefore it is rightly referred as a synthetic phase. G2 phase is the last stage of interphase where the cell organelles, say for example mitochondria, will divide and their number will double. Therefore, the answer for this question is G0 phase or G0 phase, where the cells are in non-dividing stage. So, you can call them as the differentiated cell or the permanent tissues like parenchyma are in this particular stage. So, answer is C. Second question, the kind of cell division which helps in growth, repair, regeneration and maintains the surface volume ratio is called as meiosis, binary fission, conjugation or mitosis. So, there are 4 options. The question reads like this: this kind of cell division should help in the process of growth and repair, regeneration of the lost or damaged cells, and it should also help in maintenance of surface volume ratio. So surface volume ratio has to be maintained because during S stage of interphase, the amount of DNA has doubled, which has increased the size of the nucleus, and increasing size of nucleus has put the pressure on the cytoplasm, therefore on the plasma membrane, and it is pushing them to expand. Once the cell expands, the cell surface increases, therefore the volume and surface will not match, therefore they have to be brought back to the normal, and for doing so the cell must be divided into two. Therefore the answer for this question is D, that is mitosis. So you can also include that or consider that the above mentioned properties are all the functions of mitosis or importance of mitosis. Now, meiosis is not associated with the repair and regeneration. Instead, they are associated with the sexual reproduction. Next, binary fission is similar to mitosis, but it is not a universal phenomena, and it is found only in a few kinds of cells, and it is mainly associated with asexual reproduction. Conjugation is a process observed only in case of bacterial cells, where the genetic material in the form of plasmids are shifted from one cell to another. So conjugation is a method of genetic material shifting found in bacteria. Binary fission is a kind of asexual reproduction. Meiosis is associated with sexual reproduction. Therefore, the answer for this question is D. Third question, meiosis is also referred as reductional division, as the chromosome number is reduced to half when compared to the parent cell. During which stage of meiosis does the reduction of chromosome number occur? So we all know that the meiosis, rightly said as reductional division, will reduce the chromosome number of the parent cell into half when it comes to daughter cells. And we all know that chromosome numbers are reduced during anaphase 1. Therefore, the answer is anaphase 1. Anaphase 2 and telophase 2 is similar to the anaphase and telophase of mitosis. Therefore, it does not require much explanation. The property of metaphase 1, if we have to observe, we can see that during metaphase 1, all the homologous chromosomes are held at the center of the cell in this manner. This particular equator region is now referred as a double metaphasic plate because there are two layers of chromosomes here. But when it enters into the next stage, that is anaphase 1, during this, because the kinetochore is only on one side associated with the spindle fibers, the entire chromosome is shifted to the poles. The sister chromatids are not separated. So one pole gets 2 chromosomes and another pole gets 2 chromosomes. The original chromosome number was 4 here. Now each pole is having 2. Therefore, anaphase 1 is actually reducing the chromosome number. You can come to a conclusion that B is the answer. Question 4. Read the following statements and select the right option. Statement A. Meiosis results in daughter cells which are qualitatively and quantitatively different from parent cell. B. The process of crossing over occurring during Paquetin stage forms new combinations of genes leading to variations in the offspring. Also during anaphase 1, bivalents or tetrads get separated and move to the pole without separation of sister chromatids. This question is actually the explanation for the third question. Statement B clearly explains why anaphase 1 is considered as the reducing stage. So let us understand the parts of the question here. Meiosis is referred as such a kind of division where daughter cells are qualitatively and quantitatively different from the parent cell. Quantitatively they are different from the parent cell because the chromosome number has been reduced to half. Qualitatively they are different because they have produced new combinations of gene as a result of process called as crossing over. So the answer for this particular question is A. Statement A and B are true, and B is the correct explanation for A. Fifth question. Select the right sequence of events occurring during prophase 1 of meiosis. So the right arrangement is option B. So first you can see leptotin, then zygotin, then pachytin, diplotin and diakinesis. So during leptotin you can see the formation of chromomeres on the condensing chromosome. Zygotin is a stage where the homologous chromosomes will come together or pair up by a process called as synapse. Pachytin is a stage during which the non-sister chromatids will go for the process of crossing over. Diplotin is a stage where the crossing over will almost end and starts with the terminalization. Diakinesis is actually the end of prophase 1 where left out terminalization is completed. Therefore, the prophase 1 is also completed. Question 6. Assertion. Bacteria have a short generation time of about 20 minutes. Reason for this, its binary fission is simple without requiring complex process. So both actually are true. Now assertion is, as you know, bacteria are the simple prokaryotic cells which can divide every 20 minutes, and the kind of cell division followed by them is binary fission. And the reason for assertion is very rightly said there. That is because bacteria has got a small DNA structure in the form of nucleoid. It's not very big structure. Therefore, DNA replication is completed easily. Once the double DNA is ready, with the help of mesosome will actually help in the separation of the cell. So, if mesosomes are like this, the two nucleoids or two sets of DNA get separated on opposite sides there. The mesosomes extends further, meets the opposite plasma membrane, thereby dividing a bacterial cell into two separate daughter cells. As a result of this, we can say that the right option for this question is A. Next one, we will move on to the 7th question. We are supposed to identify the right set of labelings for the given diagram. The labelings are indicated as follows. So let me get into the details of this picture. This picture is indicating the stage of anaphase. So in this anaphase, you can observe that label A is indicating this particular part here, that is the spindle fiber associated with the daughter chromosome. Label B is indicating the daughter chromosomes here. Label C is indicating the fibers which are formed between the daughter chromosomes. They are referred as interzonal fibers which will push away the daughter chromosomes. And D is actually the furrow of constriction or the process of initiation of cytokinesis. The right option for this particular question is B. Next question. At metaphase, chromosomes are attached to the spindle fibers by this. So meaning of question is that during metaphase stage, the spindle fiber should come and attach to which part of the chromosome? The answer for this question is kinetochore. Let me slightly explain this. The chromosomes can be understood as the X-shaped structures that are formed or visible very clearly during metaphase stage. Now this is a chromosome having primary constriction, secondary constriction. Primary constriction is very important, and this is having special sequence of DNA which will hold the two sister chromatids. These are the two sister chromatids. So the sister chromatids are held together at primary constriction. And secondary constriction is associated with the formation of nucleolus. Therefore, they are called as nucleolar organizing region. And please do remember, students, this is a structure not found in all the chromosomes. Few chromosomes have got secondary constriction. And if at all such secondary constriction containing chromosomes are considered, the part of the chromatids beyond the secondary constriction is referred as satellite. So satellite containing chromosomes are called as the SAT chromosomes. Moving on to kinetochore. Kinetochore is a semilunar or subspherical proteinaceous structure which covers the primary constriction, and do remember, spindle fibers come and attach to this particular kinetochore. So answer for this 8th question is C, kinetochore. 9th question, the exchange of segments of non-sister chromatids between the chromosomes of homologous pair is termed as. Answer for this is crossing over, which happens during the Pachytene stage of prophase 1 of meiosis. Now let us understand why others are not the option. A refers about transformation. Transformation is conversion of one form of cell or bacteria into another type. Translocation is indicating the exchange of fragments of chromosomes or chromatids between the non-homologous chromosomes. So it is a kind of mutation or problems associated with DNA, you can consider. Option D is chromosomal aberration. This is also a kind of mutation or problems or chromosomal disorders which will either delete the part of chromosome or any other processes will occur which leads to the problems in the working of the cell, thereby the working of the organism. So the answer for 9th question is crossing over, which means the exchange of chromatids. Next question. So this question represents the two diagrams and you have to identify those two cells respectively. So what you can observe in the diagram is that the A diagram is having still the nuclear structure and the process of condensation of chromosome has started occurring. Therefore, you can identify it as prophase. Exactly, late prophase stage is indicated. And the second diagram does not show any nuclear membrane over there, and all the chromosomes have come and arranged themselves at the center of the cell, which you call as equator. So first stage is prophase and the second stage is the metaphase. So the answer for this particular question is D, prophase and metaphase. Now the 11th question, it is regarding cytokinesis. In an animal cell, cytokinesis occurs with the formation of actin ring leading to the formation of cleavage furrow. So they have explained how cytokinesis occurs in animal cell. So animal cells cytokinesis will be formed by actin ring, which will lead to the formation of cleavage furrow. Now by looking at the two diagrams which indicates the plant cell, you have to conclude how it happens in plant cell. So in the first picture you can see the formation or association or arrangement of phragmoplast at the center of the two daughter nuclei, and in the second diagram you can see the formation of a cell plate. So options goes like this: A, same as in animal cell; B, with the formation of phragmoplast, later the cell plate; C, by the formation of membranes between cells; D, using the spindle fibers. The right option for this is B. Next one, cell A shows 5 rounds of mitosis. what will be the total number of resulting daughter cells? So the question reads like this: mitosis has occurred in cell A in 5 rounds, that is 5 times the mitosis has occurred, and now you have to predict the total number of cells that has accumulated at the end of 5 rounds of mitosis. So let us identify the options here. A option says it as 5, B 10, C 32, D cannot be predicted. Now how to calculate the number of daughter cells formed is, you have to put it as the power of 2 here. Now we know that every time mitosis results in the formation of 2 daughter cells, and how many times it has occurred, that is mentioned as a power. So 2 to the power 5 always makes it 32. Therefore total number of daughter cells occurring or found at the end of 5 rounds of mitosis will be 32. Now this was all the details regarding the 3rd unit of first year biology syllabus, that is cell. We have included its structure and function, then biomolecules and cell division has been included. Now with this particular details of all the components here, I wish you all students best of luck and thank you.